Okay, previously we talked about the basics of viral structure. Like all viruses are composed of nucleic acid. They have a protein coat called a capsid, which is made up of capsomeres. And some viruses will have an envelope, which is essentially a phospholipid membrane. So previously we talked about the structure of viruses and kind of where we find them and some classification, things like that. But today we're going to get down to what viruses actually do, and that's make more of themselves. Now, and in the process, they're going to harm host cells as well. But remember, viruses can't do this on their own. They have to be inside a host cell. So here I've said dependent on host organelles. So if we're talking about a virus that infects bacteria, which we're going to begin with, we don't really talk about bacteria having organelles, right? I mean, sometimes you consider a ribosome an organelle, but anyway, don't let that wording kind of trip you up. But viruses are going to be dependent on the host cell machinery to make new viruses. So always keep that in mind, and we'll think about maybe things that a virus needs to, encode for itself as well. There's two kinds of viral replication. So we're going to start first with the lytic replication that almost always results in the death of the host cell. Sometimes it happens quickly, sometimes more slowly. And then the other kind of replication is lysogenic replication, and that's where the virus is going to incorporate itself into the host chromosome. Okay, so again, we're going to start first with the process in viruses that infect bacteria, and then we'll talk a little bit how there's some differences when viruses infect animals, i.e. humans are animals. Okay, so 5 stages. We'll go through these here on a graphic in a minute. Attachment, entry, synthesis of new virus or new virions. Synthesis of the virus's nucleic acid, as well as then how that new nucleic acid gets transcribed and translated into viral proteins or in viral structures, and how that's all assembled together. And there's several different ways that will be several different ways of entry and release. Okay, so this is your book's graphic of the lytic cycle. It says that down at the bottom here, but I just wanted to tell you that again. Viruses that can do the lytic cycle are called virulent. Again, we're talking about viruses that are going to enter bacterial cells, so they're called bacteriophage. Okay, and we call them virulent. If they're virulent, that means that they are only doing the lytic cycle. All right, so first of all, here we have a bacteriophage, and a lot of bacteriophage have this kind of weird lunar lander type structure. Some bacteriophage are really just a capsid. Sometimes we'll see some that just have kind of this spindle and don't have these little tail fibers. So we see some different shapes for bacteriophage. Bacteriophage and any virus, they do not have motility, right? They don't have any kind of way to propel themselves purposefully. So when we talk about attachment, this is just going to be by random motion that a bacteriophage happens to land on the type of cell that it has the correct receptors for. And we do know that bacteriophage can be very specific. They may only be able to infect one species of bacteria, for instance. Okay, so just randomly happen to find the right kind of cell that they're able to infect. There are receptors, again, on these tail fibers for that specific cell. And next, the viral DNA is going to enter the host cell. So we could call that the penetration phase. It's only the nucleic acid that enters. All this capsid and the spindle and tail fibers, they all fall off and their job is done. Most often, a bacteriophage is going to enter by, let's call that a little bit more specifically, direct penetration. Again, where only the DNA enters into the cell. There's some other possibilities, but that's one common way. And it's kind of like this tail spindle here kind of contracts, and it kind of acts like a hypodermic needle or a syringe that shoots that nucleic acid, most often DNA for bacteriophage, into that host cell. This is not quite as simple as it sounds here for this penetration phase. The virus has to bring with it some protein, or it often does, called lysozyme. Sorry, it's an enzyme, lysozyme. Okay, so that will carry with it. Lysozyme, what it does, it breaks those glycosidic bonds in the N-acetylglucosamine and then N-acetylmuramic acid of peptidoglycan. Okay, so it breaks those bonds and weakens the peptidoglycan structure so that often DNA can penetrate through that cell wall. Without that, it just has a hard time penetrating. Okay, so the lysozyme kind of softens that cell wall a little bit. At this point, the virus will usually kind of digest the host's DNA and kind of take over the cell. It will transcribe and translate its DNA into its own proteins and its own structures like capsid. And those are going to get assembled. Here they call it maturation. Same thing as assembly into new virions, new bacteriophage. It's kind of a mystery how that assembly process works. We don't think there's any enzymes that facilitate that. I think it just kind of spontaneously comes together. Again, there's a lot we don't understand there. And then once those virions are mature, are all put together, then that host cell will undergo lysis. Again, lysozyme is important in this process to soften that cell wall so that the new virions can break out. Okay, so lysis, or bursting of that bacteria. If we look at kind of the pattern of virion abundance in the lytic cycle, in other words, if we kind of look at a growth curve, kind of compare it to the way, think about how bacteria grow, all it takes is one virion to enter the cell. And sometimes it could be more than that when there's lytic replication, and there often would be. But so one enters, and then you have this kind of long, you could think about it as kind of a lag period where there's no new virions being produced. And then all at once, we get a burst of those virions, when they're leaving the host cell, usually somewhere between 100 and 200 new virions, new bacteriophage, leaving the cell all at once, very abruptly. If you look at the time here, generally maybe 25 minutes. So it happens pretty quickly in a bacteria. Here's the E. coli that's undergoing lysis. Gotta kind of feel sorry for that bacteria cell just a little bit. All right, and the second possibility, there are some bacteriophage that can undergo what we call lysogeny, or sometimes we call that the lysogenic cycle of replication. And bacteriophage that can do this are called temperate bacteriophage. Temperate phage. Okay, so like I said before, what's different about this kind of replication is that the viral DNA is going to actually get incorporated into the host cell. And then, as we'll see, this lysogenic cycle actually incorporates the lytic cycle. So temperate phages are actually able to do both lysogeny and lytic cycle. All right, so here we have the phage infecting the cell. And then instead of breaking down the host chromosome, you can see here the phage DNA becomes incorporated. And it will send out some proteins that will inhibit other phage from infecting this cell at the same time. You can see how you wouldn't want lots of different phage incorporating into that chromosome because probably the cell wouldn't be viable anymore. But this step will be referred to as the prophage step. Okay, so again, that phage DNA is incorporated into the host bacteria cell. And that actually, as you would expect, a lot of times that doesn't alter the bacteria at well, but it has the potential to actually change the phenotype of that bacterial cell. And if it does, we refer to that as lysogenic conversion. And again, we don't think that happens very often, but there are a few examples that we know of. The bacteria that causes diphtheria actually doesn't cause diphtheria unless it has been infected with a bacteriophage that gives it the ability to make diphtheria toxin. Another example, Streptococcus pyogenes, that's an organism that causes a lot of different infections, but it will only cause scarlet fever if that bacteria has been infected with a phage that allows it to make this erythrogenic toxin that causes this bright red rash in humans, among other things. Okay, so we know there's some examples of when that can happen, but typically, the virus will just sit in that host cell for generations, and it will get replicated every time the host cell replicates its DNA and divides. But eventually, that viral genome can come out of the host cell, and we call that process induction. When does it do that? There's a lot we don't understand. We know that we can cause induction if we hit the bacteria cell with some radiation, with some ultraviolet light. So there's probably some kind of mechanism that when the virion senses that the cell is in trouble, that it's about ready to die, it kind of jumps ship so that it can go ahead and make more of itself. So if induction does happen, then the virion will enter into the lytic cycle, where it's going to degrade the host chromosome. It's going to make its own proteins, its own parts. Those will assemble into new virions, and then we'll get lysis releasing new bacteriophage. Okay, so what are some differences when replication of a virion is taking place in an animal cell? Or remember, humans, we're animals, so we're going to call ourselves animals here. It's pretty much the same. Okay, there's going to be a few differences because Of course, a lot of viruses infect animals are enveloped. And there, I've said in the past there's exceptions for everything, but there are a few bacteriophage that make something sort of like an envelope, but primarily, for the most part, bacteriophage are not enveloped. But a lot of animal viruses are, so that's going to change how the virus enters the cell and how it leaves it. Of course, animal cells are nucleated, so viruses that need those replication enzymes, for instance, are going to have to enter the host cell nucleus. And what do animal cells lack that bacteria have? Okay, of course, animal cells do not have a cell wall. Okay, so again, that changes, primarily the entry and the release mechanisms. Okay, so here's, they drew out here a replication of an animal virus. For some reason, they picked influenza virus, which has some exceptions to it that I'll point out. But if we think about the mechanisms of entry, and one of those is illustrated here. The one we talked about already was the direct penetration. And we said that's the way most bacteriophage will do it, but this could be any non-enveloped or naked animal virus and phage, again, which usually don't have an envelope. So again, direct penetration is just where the capsid and everything stays on the outside of the cell, and only the virus, the nucleic acid enters into the cell. There also is the possibility of membrane fusion. And of course, this is for only enveloped viruses, where, just like it says, and I don't have a picture of this, but the membrane of the virion fuses with the cell membrane of the cell. And the third way, we have actually talked about this before, is by endocytosis. And that's what's sort of pictured here. That's what the influenza virus does. And this can be either naked or enveloped viruses. So basically, the virus is going to trick that host cell into engulfing it. So we talked about receptor-mediated endocytosis before. That's actually what the influenza virus does. That's what COVID does. And this has the added advantage of not leaving the virion's membrane sitting on the outside of the cell, sort of advertising to our immune system that, hey, there's a virus inside this cell. Okay, so there's no evidence on the outside, and it helps avoid immune detection. So essentially, the influenza virus, kind of tricks the host cell into phagocytizing it. And then once that virion is inside the cell, we have an uncoating process where the membrane from the endocytosis falls off, the envelope of the original virion falls off, the capsid falls away, and we just get, the nucleic acid eventually emptied out in the cell. Now, interestingly enough, we'll talk about this more here in a minute, but influenza virus, this is an RNA virus. Just FYI, we'll talk about that virus more specifically later. But most of the time, RNA viruses, they don't have any business going into the nucleus like this because there's nothing that they need there. What you find in the nucleus are things like DNA polymerase, and an RNA virus doesn't need that. But interestingly enough, the exception here is that influenza will enter the nucleus because there's some kind of splicing enzymes that it needs, so that its messenger RNA is more likely to get read at the ribosome. So anyway, that's a long explanation. Usually RNA viruses don't go into the nucleus, but this one does. We're going to talk about release here. I've got some other pictures of that on a later slide. So talking more about that synthesis phase for animal viruses, or really, again, whichever kind of virus you're talking about, most bacteriophage are DNA viruses. There's a few exceptions to that. But depending on what kind of virus, what kind of nucleic acid is in the virus, that depends. I want to think through kind of some of the strategies, how it makes more of its nucleic acid. Whether it's a double-stranded DNA virus or a single-stranded DNA or a double-stranded RNA, again, there's some crazy stuff here that exists only in viruses. There are a lot of viruses that are single-stranded RNA. Some of them have the plus strand of RNA, and some of them have the negative strand of RNA. So remember the difference. The plus strand of RNA, this is going to function like messenger RNA. And if it's a negative sense RNA, it has to be copied to, well, not exactly copied. Let me say this a little bit better. It has to make its complementary strand to get read at the ribosomes. Okay, so hopefully this helps to illustrate that a little bit. If you have a plus sense single-stranded RNA, that RNA can immediately get read at the host ribosomes and be translated into viral proteins. Easy. But it needs to make more for those new virions that it's making. It has to synthesize more of its DNA. You can't make a plus sense single-stranded RNA from a plus sense. You have to make the complementary strand, which would be negative sense single-stranded RNA. And remember, in our cells, we don't have enzymes to make RNA from RNA. We don't do that. So the virus has to bring in its own enzyme, RNA-dependent RNA polymerase, okay, to make that complementary strand of negative sense RNA, and then the same enzyme to make more of the positive sense single-stranded RNA. Okay, so we talked about last time how the genome of viruses is pretty small. It's way smaller even than bacteria. RNA viruses in general have the smallest genomes, but one of the things that they need to bring with them is some way to copy their nucleic acid. So again, that's one of the kind of unique genes that they would have to have. Let's see. So and I mentioned this, that DNA viruses are usually going to enter into the nucleus because, again, they need that DNA polymerase 3 that the host has in order to copy their DNA. There are some exceptions to that. For instance, smallpox is a DNA virus, and it just brings everything that it needs with it. It's a much bigger virion, and so it just stays in the cytoplasm. And again, RNA viruses, usually there's nothing that they need in the nucleus. Influenza is an exception there because, anyway, they usually don't need to go into the nucleus because those, they have to bring their own enzymes with them anyway. Okay, so the point is here that whatever kind of nucleic acid that viruses have, whether it's, some kind of DNA or negative sense RNA, and don't memorize these specifics, these different viruses at this point. Whatever kind of nucleic acid they have to have a strategy to get plus sense RNA. Because remember, that's what's read at the host cell ribosomes, the plus sense RNA. Now, there are some viruses that have a little bit of a different strategy. Some of them are called retroviruses. And a retrovirus, HIV, is the most famous example of a retrovirus. There are others. They carry an enzyme called reverse transcriptase. Okay, so what that enzyme does, for instance, HIV is a plus sense RNA virus, and it will make it produces reverse transcriptase, which allows it to make a negative sense single strand of DNA, and from that, it can make a double strand of DNA. And from that double-stranded DNA, it will make more of its plus sense RNA. So it seems like a crazy thing. This virus, it's already the form that it needs to be read at the ribosomes, but instead it goes through this through a DNA intermediate to make more of its RNA. And that seems crazy. It was certainly a mind-blowing thing when that was first discovered back in, the 1970s, I suppose. David Baltimore, remember we talked about that Baltimore classification system. He did a lot of work on retroviruses. It seems crazy, but double-stranded DNA is much more stable, and that allows the HIV to integrate into the host cell. Okay, so it's one of the viruses that can do that can integrate in the host cell and remain latent. Okay, so when animal cells do that lysogenic cycle, let me do that again. Lysogeny in animal viruses is called latency. I don't know why we need a new term for that, but when animal viruses integrate into the host cell, we call that they're latent. Okay. And we're going to discuss a couple different viruses later in the semester in more detail that do that. HIV is one of them. Herpes virus is another one. And herpes virus actually makes these plasmid-like structures. It doesn't incorporate right into the chromosome, but it makes these, little plasmids. And basically every time that host cell is replicated, those plasmids get replicated as well. HIV integrates right into the host chromosome. And these animal viruses, they can just hide out there for years without any viral activity. Herpes virus can reactivate pretty often, actually. It will start replicating again, and then the host will have symptoms. Chickenpox is a herpes virus, and it can hide out for decades in a person and reactivate in a little different form, a rash that's known as shingles. Okay, but one difference here is in animal cells, when these viruses go through this latent replication cycle, it's the integration is permanent. So integration is permanent. So in other words, we get no induction into that lytic cycle for animal viruses. Okay, and that's sort of unfortunate because once HIV is in our chromosome, we don't have any way to get it out. Okay, so the release of well, talk about assembly a little bit here. The release. The assembly process takes a lot longer. So remember how long it took, we talked about it's kind of a lag period until the new virions were released from a bacteria cell. Remember, we said that was about 25 minutes. For animal viruses, it's more like 24 hours. Okay, so we get kind of this much longer lag period till we get a release. And I should draw this a little bit different. Oftentimes, the release is a much kind of slower release because animal viruses often have a membrane. Okay, there's 3 different kinds of release mechanisms. Viruses that do not have an envelope, naked viruses, sometimes they will just lyse the cell, or the other possibility is exocytosis. So that's just essentially the opposite of endocytosis. So they're kind of travel through the mature virion will travel kind of in a membrane until it gets to the plasma membrane, and then it's released. An envelope virus can also be released by exocytosis. So there are viruses like herpes virus, they actually take their membrane from the nuclear membrane of the host, and then they just travel through an additional membrane till they're released from the host cell. Another very popular way for, again, membrane or envelope viruses is by budding. So they will actually express any maybe spike proteins on the surface of the host cell, and then they will just take some of the plasma membrane with them as they leave the host cell. And when this happens slowly, it can be, often kind of a very chronic infection, right? And we would get kind of a slower number of new virions released. Sometimes that budding takes place very quickly, and in effect, it's lysing the cell because they can't lose that much of their cell membrane kind of all at once. Okay, so budding, we'll just say it takes part of the host cytoplasmic membrane. Okay, so occasionally or commonly, we may want to culture viruses, maybe to study them for research, potentially to identify or diagnose a viral infection in a patient, or certainly very commonly for vaccine development. So one method that we could use if we've got a suspension that has, maybe a lot of different stuff in it, there could be bacteria and other things present. If we would run that suspension through some kind of filter pore where some kind of membrane where the pores are small enough that only viruses could fit through. So you can see here where these pores are really small and these bacteria are too big to fit through there. So we would have a suspension, just of viruses. And of course, we can't culture viruses just on, bacterial medium. Like we can't grow viruses on nutrient agar. Okay, remember bacteria have to have cells to grow in. So oftentimes we would grow viruses in some kind of cell culture, oftentimes in kind of plastic containers that look like this, where you've got kind of a monolayer growing on the bottom of the plastic there of some kind of cells. The liquid there is the nutrients the cells need to stay alive, and then you infect those cells with the virus. Here we have some kind of maybe nutrient agar or tryptic soy agar medium where we've got a lawn of bacteria streaked on that medium. So again, if this was our plate, then we would, make a lawn of bacteria on there. And then we would add our suspension that contained the viruses. And if you wanted to count how many viruses were in that original sample, you might dilute that sample out a little bit, and then you could count the plaques. Essentially, these are little spots in your lawn of bacteria where the bacteria aren't growing anymore because they've been lysed by the virus. So this is kind of the opposite of the viable plate count, where you did this if you were in lab, where you counted the number of colonies and then were able to extrapolate back about how many bacteria were in a sample. Same thing, you can count the number of plaques and kind of extrapolate back to the number of virions in some sample. Another way that's very common to grow viruses is in eggs. You can see here that there are different parts of the egg that can be injected or inoculated there. It depends on what virus you're trying to grow, I guess, as to where the best place is. But we do this commonly to make vaccines. So there's a lot of different ways that we can make the influenza vaccine, but most commonly it's still grown up in eggs. And unfortunately, that's why it takes so long to make it, because that takes a number of months, 6 months or so, to make enough of that for what we need. So we have to decide what strains of flu that we're going to use like way ahead of time. And if we could find a way to shorten our production time, then we could be a little bit more responsive to what strains are circulating at any given time. Back to the cell culture idea, this has always been problematic in some ways because those flasks that I showed you before, they so easily become contaminated with bacteria. So we will often add antibiotics to try to limit that. This is kind of, I haven't seen one of these, but this is kind of a fancy way. You can see you've got the cells in a bag here with this fluid nutrients bathing it, and this is more of a closed system that would be less likely to become contaminated. There's different kind of cells that we can use to grow viruses in. If it's a primary cell culture, basically these are cells that are prepared from animal organs or tissues, and they are going to have contact inhibition, which just means that the cells will stop reproducing when they come in contact with each other. So with this kind of primary cell culture, you have to be constantly transferring cells into a fresh medium. And there's kind of a limit to how many times you can successfully do this until the cells will just die. So a lot of times we like to use a continuous culture where we use a different kinds of cell. We use cells that have been harvested from tumor cells. The most famous ones are HeLa cells. The tumor cells have lost their contact inhibition. So basically, they can be grown in suspension, and they'll pile up into clumps, kind of like tumor cells were, and they'll just keep growing and growing and growing indefinitely. So that's kind of a handier way, I guess, if you're studying viruses. So that's pretty much what I want to talk about for viruses. Just visit the notion briefly here. There is kind of a debate over whether you would consider viruses alive or not. And people will continue to argue about this. If you look at the 4 characteristics of living organisms and you think, well, do viruses check all these boxes? They definitely don't check all these boxes. Do viruses grow? In other words, do viruses grow into adult viruses? No, they are just assembled, like a assembly line at GM. Do they reproduce? Yes, but they have to be in a cell. Now, there are some bacteria that can only reproduce in a cell as well. Are they responsive? In other words, do they respond to stimuli? This is the one that people will argue about. We'll say no, although somewhat more arguable. You could say, it depends how you define this. You could say, well, they do evolve over time. Certainly we've seen that with COVID. They mutate and evolve over time. But in general, again, they don't respond to stimuli. They don't move in any, quicker fashion than that. Do they have metabolism? No. Okay, they can't take in nutrients. They don't make ATP. So, again, because they don't tick all these boxes, most people would say no, but some people like to argue about that. And before we move on, I'd like to show you, I don't think I posted this, but this is an article that was in the Wall Street Journal about 5 years ago or so. I don't think I wrote the date down here. But it's always interesting when you read about science subjects and how, a non-scientist might write it up in the media. Because here this talks about baby phages taking over the bacterium's body. But this is a really interesting article, how bacteriophage are being used to fight bacterial infections. It's a huge problem for burn victims, especially because they've lost their skin. They get really bad infections really easily. So they did this whole study where they used bacteriophages that were specific for Pseudomonas, which is a very common hospital-acquired infection, especially for burn victims, and dipped them in these bacteriophage that would kill Pseudomonas, and they had a lot of success with that. Again, this hasn't caught on in this country because people are wary of using viruses, and you just have to make sure that they're not going to respond in a way that we don't, understand. So, promise for the future, though, potentially. All right, let's, to finish out our chapter 6 here, let's talk about another thing that's really small that can kill you, and this is prions. Prions, this is a term that was coined by Stanley Prusner in 1982. So this is fairly recently that we have understood what prions are. Prions is just kind of an abbreviation for proteinaceous infective particles. Okay, so this is a really mind-blowing thing that there's something that can make you sick that's just a protein. Okay, so there's no nucleic acid here. So the question is, how do they make more of themselves? If there's no genetic material involved, how can a protein make more proteins? Well, we think the way this works, there's this protein that we call prion protein, or PRP. It's a membrane protein that's found in all mammalian cells, and we don't really understand the function of it or why we have it. But we do know that when the variant form of this prion protein is exposed to the normal form, then the variant form, which it's variant because it's folded a little bit differently, when it's exposed to the normal form, it will cause the normal form of the protein to also misfold. So it's kind of like, if you've ever heard the expression, one bad apple ruins the bunch, that's the idea. Okay, so here's this cellular PRP. That's the normal kind that, again, is in all of our mammalian cells. When it's exposed to the misfolded version, there's a few more of these beta sheets rather than alpha helices in the misfolded form. And again, when it's exposed, it will cause the normal form to misfold. And the more beta sheets there are, that causes the proteins to clump together, and eventually that causes the neurons to stop working and die. So, some examples of prion disease, these are all degenerative diseases, and they are always fatal. Of course, we're working on therapies for these, but we haven't made much headway the last time that I checked. So you've heard of some of these. We call them transmissible spongiform encephalopathies. I always have trouble saying that word. If you basically all of them, if you look at normal brain tissue, it looks like this. Any spongiform encephalopathy will form these plaques where the neurons have died and have stopped working. So some examples, bovine spongiform encephalopathy, you know that as mad cow. Okay, so this, we still see cases every once in a while of this, but this really made the news every night in kind of the late 80s, early 90s. It was kind of centered in England. At that time, it was the practice to feed animals. A huge component of animal feed was dead and ground up dead animals, like all parts, bones, muscle, brain, everything. So we learned that we have to stop doing that because that was how mad cow was being transmitted, the brain tissue of a cow that had died of mad cow, eaten by another cow. And if you Google mad cow with mad cow, it's really disheartening seeing a cow like stumble around and it's just kind of frightening. So back in the early 1990s, I spent a few weeks in the summer in England, and all I heard was, don't eat hamburger, don't eat hamburger. And at the time I thought, well, that's crazy. Of course, hamburger is cooked, so what's the problem? Well, prions are not killed, we could say, or inactivated by regular cooking. You have to heat prions up to 480 degrees Celsius, which I think is like 900 degrees Fahrenheit, for 4 hours to inactivate them. So normal cooking is not good enough. And back at that time, the early 90s, I think about, of course, lots and lots of cows had to be sacrificed, but I think about 200 people died of mad cow disease at that time. Scrapie is just basically mad cow and sheep. Again, it makes them act oddly. They will scrape up against a fence until all their wool is worn off. Chronic wasting disease, that is in deer, elk, and moose, and we have had cases of that in Michigan. Luckily, And how is that spread? We think through contaminated body fluids and tissues, maybe even drinking water, or maybe through food. There haven't been any cases of humans contracting it from deer, elk, or moose. Not yet, anyway. So that's the good news. In humans, what do we call this? Variant Creutzfeldt-Jakob is when humans ingest contaminated meat. And like I said, there were about 200 humans that died of variant Creutzfeldt-Jakob by eating a cow that had died of mad cow or had mad cow. Then Creutzfeldt-Jakob disease is the genetic form. So we think it's potentially, a rare genetic mutation that can cause that. Kuru is no longer circulating. This was a type of ritual cannibalism that's in Papua New Guinea. It was, for the natives there, it was a form of respect when your relatives would die to eat parts of their body. And the brain was, that was the most coveted thing to be able to eat. So they have been taught to no longer do that. And so, again, Kuru is we don't have any more cases of that anymore. So prion disease, it can be inherited, again, a rare genetic mutation, what we would say kind of sporadic, where that misfolding arises from a rare random change, or infectious, meaning a person has had some direct exposure, like eating a sick animal, or potentially some transplanted tissue. Very difficult to, again, disinfect medical instruments and things like that. So. So prions, really interesting. We won't spend any more time there. I've got one practice question here before we end our discussion of viruses. So I would stop your video for a second and give it a try. This is just, remember, a term that we talked about earlier, lysogenic conversion. What is it? The correct answer here is B. The removal of phage DNA from the host chromosome, remember, that's called induction. This is true. This happens, but it's just part of not only penetration of the virus as well as when the virus leaves the host cell before it lyses it. And I don't know if this has a name, so... Okay, thanks for listening.